Okay, Bio 100, this is your abbreviated lecture for Monday, March 9th. As I pointed out in class, I'm going to be at a meeting, and we are going to make a little bit of progress here in Chapter 9. Remember that we do have an exam that will be held this Friday, or this upcoming Friday, which is the 13th, and this is part of the material that will be on that exam. Okay, so we got to mutations today in class, and we said that a mutation is a change in the DNA sequence. And when we change the DNA, then we're going to change the protein, or at least most likely we are. In some cases, we actually don't, as you'll see in a minute. But it's very likely that if we do change the DNA, we're going to have a change in the protein that's produced from that particular change. And remember, what really matters is the protein. If we change the protein, we change the organism. So there are three possible outcomes for a mutation, a change in the DNA. One possibility is there's no change in the protein. We'll explain how that could be in just a moment. A second possibility is that we produce a non-functional protein, a protein that's different and doesn't do its job. The third possibility is we produce an entirely different protein that maybe does its job, or maybe it does it better, maybe it does it worse, or maybe it does something else entirely. So we'll talk as we move forward here about what might lead us to observe any one of these three possibilities. Now, when we talk about mutations, we typically lump mutations into the following categories. The first one that we have here, and each of these could either be as we see on this slide, could cause no change in the protein, a non-functional protein, or a different protein entirely. So one type of mutation is a base substitution, and otherwise known as a simple substitution. We simply take one base and we trade it for a different one. You'll see the cursor, there it is, moving around on the screen, and so you can see in the top we have our original DNA sequence here, and then on the bottom we have our mutated DNA sequence. And so the original DNA sequence is ATC, GCA, and in the bottom sequence we have ATC, but then we trade guanine for thymine, and so it reads TCA. And so a simple substitution may or may not cause a change in the actual DNA sequence, as we'll see on the next slide. The second possibility for types of mutations, the second possibility is a frameshift mutation. And so in a frameshift mutation, we add or delete a base in such a way that the reading frame is actually shifted. For instance, on the bottom down here, we read, remember that the RNA is read in sets of three, referred to as codons. So down here we read ATC, GCA, AAT, and CCG. Now, when the mutation occurs, we insert one thymine, a single base, into the DNA. So now we read ATC, but instead of reading GCA, we read TGC. And then that means that each codon after that is changed. And so you can see that AAT is supposed to be a codon, but now that equivalent codon is AAA. The next one is supposed to be CCG, but now it's TCC. And so frameshift mutations would be expected to have a great influence on the protein as a result of the fact that you don't just mess up one codon like you did in the case of a substitution, but you mess up all of the codons after the point at which the base has been added or deleted. A deletion can also add to a frameshift. Of course, you could have an addition or deletion in a multiple of 3 that wouldn't cause a frameshift, but anything other than that will cause this frameshift mutation, which we would expect, again, to have a much more profound effect as opposed to just a simple base substitution mutation. And I think you can see, hopefully that is clear as to why that is. Alright, and so here we see the effects of a mutation in this diagram from our text. So on the left-hand side we see a normal DNA sequence. So you see the double-stranded sequence. We are reading the top sequence. Remember in class today we said that you either read the top sequence or the bottom, but you don't read both. And so here we take the top sequence, we turn it into messenger RNA. T becomes A becomes U, C becomes G. That's our start codon, which codes for methionine. And then we have our protein down here, methionine, and so forth. We see the amino acids connected together. And we have our normal functional protein. Remember, it's shown in a globular shape because there are probably over 1,000 amino acids that make up the protein, and it's all twisted and bent and curled around on itself to make the protein that we see on the screen. Over here, on the right-hand side, we see a mutated DNA sequence. And so it looks like in this case we are trading thymine, T-A-C-C-T, for cytosine. So you see on the top we have changed that thymine for cytosine. That becomes a guanine then on the bottom, so the bottom changes as well, but that doesn't especially matter because we're reading the top sequence and making RNA out of it. So now in the RNA codon, instead of having GAU, which codes for a particular amino acid, over here we have GGU. So we've traded amino acids. We now have glycine at that particular location on the protein, and that means the protein is different. So interestingly, we talked in class about how proteins curl back around on themselves, and in that curling process the amino acids at different places on the protein interact with each other. And so if you trade one amino acid for another, it's quite possible that those two amino acids will not interact with each other. For instance, they won't form a hydrogen bond perhaps, and that means that that particular part of the protein falls apart. I'll have to illustrate this in class as well on the board, but it falls apart, and so we end up with a protein that doesn't work as a result of that change in the sequence. And so this is an example of a situation in which a change in the DNA sequence does cause a change in the protein, and it produces the worst case scenario, which is a non-functional protein. So here we have some other examples that are very useful. So look, if we go back to, well actually this is a totally different example. We don't need to go back to the previous page. So different figure from our text. So here we're taking this sequence of DNA, I'm pointing with the cursor now, this sequence of DNA, turning it into messenger RNA, and then we're turning it into our normal protein sequence that we see here. Well, we said that there are three possibilities for what can happen to a protein when you have a mutation. One of which is that nothing happens. It has no effect on the protein, and that's what's happening here. So look, sorry about that. Look what we've done here. We have ACT, AAC. Now compare this to the original sequence, ACT, AAT. So we have taken thymine and we have traded it for cytosine. So we have changed the DNA sequence, but watch what happens here. We have ACU and AAC are found in the RNA, and if we look up here at our original RNA, we have ACU, AAU. Now you'll remember, to understand what's happening here, you have to think back to the table. In fact, I'm going to scroll back to it right now, this table, and remember that there are multiple amino acids that code there are multiple codons that code for particular amino acids. In this case, we have asparagine. And so look, what we see is that AAU and AAC, what amino acids do they code for? Well, they both code for asparagine. And so that makes sense then. So if we go back here, we can see that we have changed the messenger RNA sequence, but since we changed AAU to AAC, both of those code for asparagine, so there's no change to the protein at all. Now this means the mutation truly is silent. There would be no effect on the organism at all. You would not know the mutation is there. It's all about those amino acids. As long as the amino acids remain constant, then there is going to be no change to the protein itself. Now the other example here is a frameshift mutation, if we go to B. And so in the frameshift mutation, in this case, we're supposed to have this particular amino acid here, but it changes to a stop codon. And so this is devastating for the protein. The frameshift mutation resulted in the production of a stop codon. And the protein, who knows, there may supposed to be another 600 amino acids after this particular location, but now there are none. So it's almost guaranteed that this protein is going to be completely non-functional as a result of this mutation. Now the last possibility that we saw in the beginning of our little discussion here, well, let's just go back. The last possibility we saw was that you end up with a different protein, a completely different protein. Well, that's possible, and that would not be a neutral mutation, but that would be a protein that is completely different and has a slightly different function. It's most likely that you would take a protein that performs a certain function in the body and modify it just a little bit so it does the same function but does it better or worse than it did previously. You know, what's a good example? Well, the sickle cell mutation that causes sickle cell disease formerly known as sickle cell anemia, is a great example of that. In that mutation, you have mutated hemoglobin proteins, which are found in your red blood cells, and there are two effects that are brought about by that protein. It's a different protein, and so it still carries oxygen, but it does not carry oxygen quite as well as does the original protein. For instance, this has been in the news, I think this was last year, but Willie Cauley-Stein, it's been pointed out, the UK basketball player on this year's team is a carrier for sickle cell anemia. So he has some of his hemoglobin proteins have this mutated state. And so sometimes, these news articles said, he gets a little bit more winded than do other players, which would be consistent with the condition. Those hemoglobin proteins behave differently under low oxygen conditions and are not quite as effective in the transfer of oxygen through the body under low oxygen conditions. It's no big deal, but it doesn't cause any serious consequences, but it's an example of a different protein that behaves a little bit, does the same job but a little bit differently. Now, we can also point out, this doesn't apply to Willie Cauley-Stein, but that different protein that he possesses also makes the red blood cells immune to malaria, so he cannot get malaria, and so that's why that mutation is so common in the world today. And I think we've talked about that before, and we will talk about that again later in our class. All right, and so that's a brief overview of mutations.